Today we'll be looking at the functional anatomy and physiology of nervous system. Okay, so nervous system is an organ system in our body, like any other organ systems, digestive system, respiratory system, and kidney, nephrology. So its main function is control and coordination, and control and coordination of different body organs to achieve one simple purpose. It needs to receive signals, process them, and send out a command. Okay, so if you look at that, if you divide this organ system into its constituents, let's say here is our organ system, that is nervous system. It is made up of organs, which in turn is made up of tissues, which in turn is made up of cells. So different cells, they do different functions. The main cell that is doing the job in nervous system is, the functional unit is a neuron. And neuron alone cannot do all the functions. It needs some supportive connective tissue cells. Namely, you have astrocytes, oligodendrocytes, ependymal cells, and finally you have microglia. Okay, so out of all these cells, neurons are the functional units, basic functional units that does this job of receiving information and processing it and sending out a command. So essentially, what kind of information? So the neurons that are taking in information are called sensory nerves, and they send in information to the central nervous system. And the central nervous system basically is brain and spinal cord. Okay, all the sensory input is going into the central nervous system. What kind of sensory inputs? Okay, we have our 5 senses, right? Touch, taste, smell, audio, and video. And apart from this is from external environment. This is external environment. See, somebody touches us. So what are all the things that we are feeling? We are feeling like there is a touch sensation, there is also a temperature, there is also pressure. Somebody is pressing like this or just touching like this. So you need to have the somatic information and also the chemical information, right? The chemical information from our internal environment. What is blood pH like? So that should be immediately conveyed to the central nervous system so that it can take an action on that. And what else? It need to track the pressure in blood vessels so that either the blood vessels can dilate or constrict to keep the pressure normal. And chemoreceptors, any changes in carbon dioxide concentration in blood, that needs to be immediately reported. All these chemical information from the internal environment. These are all special senses. Other senses like touch, taste, touch, pressure, position, and some chemical information from our blood and changes in pressure, etc. All this information is going into the central nervous system to brain through spinal cord. From there you have sensory, and here you have motor. That means what? From CNS, a nerve is coming and talking to some of the organs. In this case, whether it is voluntary, it is mostly skeletal. Okay, biceps is a skeletal. All the muscles in our hand are skeletal muscles. That means they are under voluntary control. Right? If I want, I can lift, but I cannot say the same thing to the smooth muscle in my digestive system. So, and there is some autonomic nervous system, both sympathetic and parasympathetic, which are sending signals to smooth muscle and some glands. Right? For example, salivary gland is one, adrenal gland is one, pancreas is one. So this internal commands are sent through autonomic nervous system by both its branches, sympathetic and parasympathetic, and motor functions to skeletal muscle is by these motor nerves, somatic nerves. So this is basically the overview of the nervous system. Now we try to understand what are the different units in that. So neurons they come together and form a nerve. So shall we call a nerve fiber or a nerve a tissue? Yes or no? What is a tissue? Group of cells coming together and doing the same function. Right? Group of cells coming together and doing the same kind of function is known as nerve. So, and this nerve tissue, they come together, all the nerves they come together and they form different organs. One of the organ is what? Spinal cord, cerebellum, brainstem, and finally we have cerebellum. So these are different organs from this nervous tissue that are formed, and finally this is becoming both central nervous system and peripheral nervous system. So or collectively the nervous system. So this is how it is structured. Now we go one by one. Okay, what are the functions of a neuron? What is the structure? How an impulse is generated and propagated. So the way the neuron receives signals is by generating an electrical impulse and releasing a neurotransmitter. So we see the basic functioning, structure and functioning of neuron and how they come together. Which part of the neuron is coming together to form a nerve? What are the components in a nerve? And then how these nerves are coming together and forming a spinal cord. What are different parts of spinal cord? What is grey matter? What is white matter? How they are formed? Then what is the cerebellum doing? Which kind of function of the nervous system is it taking care of? What is there in brainstem? We all know brainstem consists of midbrain, pons and medulla oblongata. And finally, what is cerebrum? How grey matter and white matter are distributed in all these organs. And finally, how the nervous system is able to accomplish its job of control and coordination. Okay, now let's start with the most favorite of the nervous system, that is our neuron. Neuron is the functional unit of the nervous system. So it has got a cell body with a nucleus and it has got an axon, and it also does have dendrites. So they are extensions of the cell body, just like any other cell. This neuron also contains nucleus, mitochondria, all these things it does have, and its function is just like any other thing. Only thing that it is capable of is generating an impulse. Okay, so impulse is the way the neurons communicate with each other and other cells. Impulse generation and impulse transmission. So how this neuron is able to achieve that? So obviously it should have an impulse receiving structure that is mainly done by our dendrites. So these are dendrites, and most importantly they work as receptors. They receive information and from there the cell body processes it and transmits through the axon. It's always one direction. So this is axon, and of course this is cell body, right? Now if you look at the axon, this is called axon hillock. From there it is an elongated process, right? And it ends up in, again, this small processes, okay, and they are called presynaptic nodes. They end up in a synapse. So there are two ways in which an axon can end its processes. One is either it can communicate with another neuron, they can talk to another neuron, or it can talk to any other effector organ. For example, it can be a gland, or it can be smooth muscle or skeletal muscle. If it is talking to a muscle, it is called neuromuscular end plate. And if it is talking to another neuron, it is called a synapse. It is called a synapse. Now, just to recap, neurons are the cells in nervous system. That's the basic functioning units. They have dendrite, cell body, and axon. Axons end in either talking to another neuron or to an effector organ, where a command is passed on effector organ, right? So dendrites work as receptors. That is where an impulse is generated and is passing through the axon. Impulse is generated and transmitted to neuron. Now, to pass this information, we have something known as a concept called action potential. So the action potential, how do we explain this? Let's say this is the axon, okay, and we have in our body, this is extracellular environment, fluid. This is inside axon, that is intracellular, okay. Now the predominant cation in extracellular fluid is sodium, predominant cation, and in intracellular is potassium. Okay, so usually the concentration of sodium is high on outside, so it maintains a positive charge on outside. Okay, positive charge on outside, and we have a negative charge inside the axon. Okay, now what we call this condition as? This condition is known as polarization. Polarization. Charges are distributed differently from inside when compared to the outside. This is called polarization. It is also known as resting membrane potential. Right? Now we stimulate this. We stimulate this axon or cell body. Now, if we stimulate here, we are changing the negative charged environment inside the cell, and here we have positively charged atmosphere outside. Now, here when we stimulate this, what happens is sodium from outside moves in, creating a depolarization. Creating what? A depolarization. So there is a sodium potassium pump which is activated, and all the sodium are sent in. Now the negatively charged environment is suddenly becoming positive. Okay, suddenly is becoming positive and the electrical balance here is lost. Okay, so here the homeostasis is disturbed and the cell has to come back to the normal state. So this impulse from here, it opens another gate, another gate, another gate, another gate, like this. This charge, electrical charge, moves in this direction. Moves in this direction. Now, this is where we need to understand the importance of nodes of Ranvier or Schwann cells. Okay, we all know that neurons or the axons are covered by myelin sheath. What is this myelin? How it is helpful in conducting an impulse? If it were to change the electrical activity, it is a slow process. First this has to open, slowly this is moving in this direction. Okay. Now, what if signal after reaching a time from here it can jump to here? So if you have an insulation here, so the change is happening. So essentially what is happening here? The signal is jumping from one node to another node because of the presence of this myelin sheath produced by Schwann cells. Now how it happens? Here the change has happened and from here the channels cannot open. Only here it can open. So the movement of impulse is fast. So it is doing somersault. It is doing somersault compared to this normal depolarization, or this is also known as action potential. An action is initiated, right, with a change in ionic balance inside an excitatory neuron. We are invoking an action potential that is transmitted through the length of axon if it is myelinated in a somersault fashion. That is why it is called saltatory conduction. Okay, this is possible only because the axons are covered by myelin produced by the Schwann cells. Okay, in this way you can propagate the impulse very quickly. So this axon is also called, another name for this is, it is a nerve fiber. It can form a single nerve. Okay, it can form a single nerve. Many of these nerve fibers come together, group together and are form a bigger nerve. They can work as a nerve. So there are two types of axons or nerves. One is myelinated and second one is unmyelinated nerve fibers. So in which kind of nerve fiber the conduction is fast? Myelinated is always fast, almost like 150 meters per second, whereas unmyelinated it is very slow, hardly half a meter per second. Okay, now let us look at the structure of, okay, just to recap, when a cell is excited, the changes in ionic potential lets the impulse pass through the axon. If it is myelinated, it is called saltatory conduction because of the somersaulting way of propagating an impulse. Impulses need to be generated if it is activated, right? It is stimulated. The impulse is generated and is going in this way. For that, myelinated, they carry fast. The way it happens is sodium moves in and changes the polarity called depolarization or invoking an action potential. Now, finally, how is this ending? So, Let me draw it here. This axon, we have, right? Let's say this is an effector organ. This is an effector organ. What they do is, if you enlarge this and see properly, this process, the presynaptic knob, right? After passing of electrical charge in this direction, okay, it has got vesicles inside. What do they have? They have small neurotransmitter vesicles inside. So they are released into this synaptic cleft. They are released, and the effector organ has got receptors for that. They have receptors for these neurotransmitter. There are different types of neurotransmitter. So neurotransmitter is self-explanatory. It is transmitting an information and it is transmitting in nervous system. Now, these neurotransmitters, what are they? For example, adrenaline is a neurotransmitter, acetylcholine is a neurotransmitter, GABA is a neurotransmitter, serotonin, all of them, dopamine, they all work as neurotransmitter. Depending upon the effector organ that is receiving the neurotransmitter and the kind of neurotransmitter that is released, the effect will be different in different organs. Right? So, just to understand the function of a neuron has got dendrites which work as receptors. When an action potential is invoked or when it is stimulated, the stimulation moves in only one direction. If it is through a myelinated nerve fiber, then the movement is quick, ending in a synaptic cleft with the release of a neurotransmitter. Now, this is the basic functioning of a neuron, right? A small note on how actually, if you look at the Schwann cell, let's say this is a Schwann cell. Here we have nucleus of Schwann cell. The way, see, this is our neuron. This is our axon, and the way it forms is, it goes inside and forms a sheath around it. It forms a sheath around it. So let us draw it here. The Schwann cell is like okay. Now it slowly moved inside, and here we have our axon, and of course here we have our nucleus of the Schwann cell. So what are they? This is our axon, this is our Schwann cell, and this is nucleus. Same thing, this is also nucleus, and here is our axon. And deep inside here we see the myelin. Myelin is produced by these Schwann cells, and this is, if you take a cross section of the axon, that's how it is, it looks, and these gaps are called nodes of Van Weyck. Okay, right. So this is the basic structure and function of neuron. The function of nervous system is to receive signals, process them and send it to. Now we know how a signal is initiated through a receptor, change in electrical activity ending in a chemical. So it is neurochemical transmission. The way a nervous system works or functions is through both electrochemical transmission, changes in electrical balance of the cell, as well as finally resulting in release of a neurotransmitter, which is a chemical. This is a chemical, right? We have seen the functioning of neurons, how an impulse is generated, how it is transmitted, what is saltatory conduction, and what is myelination and what is demyelinated or unmyelinated nerve fibers. Now it's time that we look at other cells that are there in the nervous system. We discussed, apart from neurons, okay, under cells category, apart from neurons, what do we have? We have astrocytes, right? And we have oligodendrocytes, ependymal cells, and finally microglia. Okay, now let us look at the nervous system. If we look at the nervous system, let's say these are neurons, okay. This is all neuronal tissue. Okay, these are dendrites. One important thing we need to understand is that they never directly communicate. These ends, they never meet. Okay, so there is always a gap, and that gap is called synapse. Okay, now this nervous tissue needs nutrition, that means food and oxygen. So there must be a blood vessel that is coming in. Okay, if a blood vessel or a small capillary that is coming in, and since nervous tissue is very sensitive, the constituents of the blood that are coming inside, okay, should be very closely watched and guarded. It cannot let everything that is there in the blood to enter into our system, because it can harm the sensitive neuronal tissue. So what we need to have here, we all know that capillaries are made up of endothelial cells, right? Let's say this is an endothelial cell. So there must be something in the capillary, around the capillary, to prevent from blood constituents to come out and harm the neuronal cells. So usually, what do we call these cells who scavenge this thing? So we have macrophages, right? These macrophages are called our microglia. These are microglia. So they have to patrol the constituents. If at all any bacteria or virus, something is entering into the system, any harmful substance, this will watch and immediately try to control that damage. So that is the function. So this is like protection. Microglia are there to protect the neuronal tissue. Second thing, there has to be a strong border, right, around this capillary vessel. So we have something known as astrocytes. What they do is they cover up. They cover up this area. completely. Okay, astrocytes are star shaped, so they cover this entire area. If you take a cross section of this, how does it look like? It should look something like this. This is a single cell thick endothelial cell lined capillary, and we have a protective macrophage around it, and finally the food processes of astrocytes. Together they form a barrier, so that the constituents are not coming freely into the neuronal tissue. So these are food processes. They are covering the blood vessel completely. Okay. So what are they doing? They are creating a barrier. Astrocytes creating a barrier known as blood-brain barrier. What is the function of neuron? Neuron is like initiating, generating and transmitting an impulse. This is the main function, right? So to receive information and to give commands, this is the main function of our neuron. So rest of the cells, they provide support, the connective tissue. These oligodendrocytes, they provide myelin-like substance in unmyelinated cells outside. Ependymal cells in ventricles, they produce some CSF, cerebrospinal fluid. We'll see more of cerebrospinal fluid later. Once we cover the spinal cord, brain stem, and finally study the cerebellum, we'll see what are meninges and what is the cerebrospinal fluid and all this stuff we'll see. Okay, so these are basically cells. So you will find a lot of oligodendrocytes here inside in the neuronal tissue. They provide a kind of support, right? They form the interstitium inside the neuronal tissue. These are oligodendrocytes. We have astrocytes here, microglia here, and ependymal cells usually they cover the lining of the ventricles. We'll see what ventricles a bit later, and also the lining of spinal canal. Okay, this is about all the players, all the types of cells. See, it is very important to understand the anatomy. Why we need to study is, if neurons are damaged, they cannot regenerate. Okay, but neurons as in cell body, if there is damage to axon, it can grow back. But the neurons cannot regenerate or divide. They cannot grow in numbers. So they need to be protected. If we understand the function right, that astrocytes is providing blood-brain barrier, which makes logical sense, and there has to be somebody who is producing CSF. There should be somebody who is protecting the patrolling the blood vessel that is coming in, because blood is the communicating mechanism through which the whole body, the total biochemistry of our blood is communicating to all different kinds of cells. Some of them are harmful. So urea, uric acid, some of them they should not enter the toxic substances. For example, drugs. Okay, only oxygen, glucose, and things like alcohol are readily absorbed into the, I don't know why brain likes alcohol, but alcohol freely enters into the brain, and some of the drugs, they are not allowed through blood-brain barrier. They are stopped. Big molecules, they are not able to come. So these are basic players when you are studying nervous system. You get them right, then it is all about organization. It's all about functionality. How these different cells are coming together to achieve a function of receiving information and how it should come. See, if we look at our body, there are different kinds of receptors. Say, we need to know about the touch, pressure, temperature, all these things from our hands, from our body, from internal system, all of them are collected by the receptors and are going to where? And are going to through spinal cord, through brainstem into our cerebrum, central nervous system. So from cells, now we move to a tissue. What exactly? We know neuron, and we need to know about a nerve. Okay. So this is a small nerve. Okay, one axon, and axons like this come together and form a bundle. Okay. These are all axons. And bundles like this with lot of axons in it, they come together and finally form a big nerve. So this is the structure of a proper nerve. Because we have to collect a lot of information from body is coming through these nerve fibers which form bundles, nerve bundles, and finally form a big nerve. So this nerve need to have its own food supply, is it not? This axon is a living tissue, right? It also needs oxygen and food. So we have our arteries, our small capillaries. Along with an artery, what should be there? A draining vein. Okay. This is a draining vein. This is the structure of a big nerve. So it is made up of axons. Axons form small bundles, and finally this forms a big nerve supplied with blood. And now, where is this going? All the nerves, they have to collect to one place, come to one place, and from there they go to brain. So nerve is one such tissue, and the next organ from cells we are going to a tissue. We have seen nervous tissue. What is it made up of? And from here we are going into the specific organs in nervous system. So all the nerves, they end up in spinal cord. Now we look at the structure of spinal cord. Okay, let's say this is an individual, right? And different kinds of neurons are going from here. Different neurons from all parts of the body, all the receptors, they are collecting from hand, right? All of them are collecting and forming, entering where? They are entering into the spinal cord. Okay. They are entering into the spinal cord. All these are nerves. Okay. All these are nerves. Now we have to look into the structure. What is the cross section? How does the cross section looks like? So from there, finally it is going into the brain, right? This is brain. We have brainstem. So this is the connecting spinal cord, right? Now we'll see the structure of the cross section of the spinal cord. So few terms before we go into the spinal cord. One is we need to understand what is grey matter and what is white matter. And another few words that we need to understand what is gray matter and what is white matter. Okay, so the neurons are usually unmyelinated, is it not? The neurons are unmyelinated, right? So you see myelination only in axons. Right? So gray matter is usually made up of all axons because they look white. Gray matter is made up of predominantly of axons. I'm sorry. Gray matter is made up of neurons. White matter is made up of axons. And gray matter is predominantly by neurons. Okay. So now we know that in spinal cord cross section we have gray matter here. This is all gray matter. Okay. Now we are taking a cross section here. So the nerves are coming and entering into these paths. Okay. So this is posterior horn and this is anterior horn. This is where the nerve root forms. They come together and form the nerves. So this is how it looks like, the spinal cord. If you take it down, so this is how it is formed. Okay. So the same thing will come down here. That's all. The cross section of a spinal cord. Now to understand this better, we need to understand two important concepts. Just like gray and white, we have what is known as nuclei and ganglia. Nuclei is collection of neurons in CNS, right? And ganglion is collection of neurons in PNS. So what is PNS? Up to this part, the nerves, they come together, join and go out as a spinal nerve. Okay. So they come out and go out as a spinal nerve. So these nuclei, if group of neurons stay somewhere before entering into the spinal cord, we call it as ganglion. So this particular part is collection of nerve cell bodies or neuronal cell bodies. Now let's say from here information, let's say from the skin. This is the skin. From here, the neuron is carrying an impulse, particular information. So this is coming to the cell bodies and is relaying it into another let's say neuron. This information from skin, let's say there is a small prick, this information is carried by the posterior root of the nerve and this is called posterior root ganglion. Okay, so you have this posterior median. This is white matter, lateral white matter, posterior white matter, anterior white matter, again lateral white matter. It is self-explanatory. So, what are they made up of? Ideally, what are they made up of? They are all made up of, we saw those nerves, right? As long as it is outside, an axon is outside. Okay, or a nerve is outside, we call it as nerve. Okay, next, another word we need to understand apart from nuclei and ganglion is, let me put it this way. Okay, so you have nuclei in central nervous system, which is collection of neurons, and ganglion is collection of neurons in peripheral nervous system. Group of cells stay together outside of central nervous system, they are called ganglion. This is posterior horn of the nerve and this is the anterior horn and this is posterior root ganglion. Okay, if at all the same collection of cells that are there in central nervous system, we call it as a nucleus. Okay, don't confuse with the nucleus of a cell. There are different nuclei, group of neurons collectively do a function that is called nuclei. Okay, and another most important thing, what is the difference between a nerve and a tract? Right? Nerve, we call this particular axon a nerve provided it is only there in peripheral nervous system. Got it? They only in peripheral nervous system. Let's say this is our guy again. Okay, and we have our spinal cord here and going up into the brain. The nerve, as long as it is staying outside of central nervous system, central nervous system is both brain and spinal cord. The moment this nerve is entering into the spinal cord, it is entering into the central nervous system. So, from there onwards, these are called what? Tracts. Tracts in central nervous system. Okay. Now, just to recap, what is grey matter? Grey matter is collection of neurons come together, unmyelinated neurons and axons, because they don't have any white colored myelin with them, so they appear grey in cross section. So we call this as grey matter. Okay, and outside this area is full of tracts. Don't again get confused with nerves. There are no nerves in central nervous system. Central nervous system is both spinal cord and brain. So there are no nerves. The nerves, they end here the moment they enter into the spinal cord. So these are tracts. They are called tracts. Okay, they are going up like this into the brain. So these are collection of axons which are called tracts in human body. So this is the structure of spinal cord. So what is spinal cord doing? All the nerves that are coming from our body, enter into the spinal cord. From there it is going up, it is taking the information up. So another important thing that we need to understand about posterior root and anterior root is most of the anterior root neurons are motor in nature, right? So this is a cell body. This is the axon. It comes out through anterior nerve root, and it's going into an effector organ. Maybe this could be a muscle. This is small muscle. Okay. So what is motor? Motor is movement. Motor means movement. And it is effector. What is this? It ends in a neuromuscular end plate. Okay. Neuromuscular end plate with a muscle. This is the muscle. This is the nucleus. Right? So this is motor, anterior motor, and posterior is afferent. This is sensory in nature. Okay. Sometimes what happens is the connection could be after receiving the information from here going into this place. We know that there is a spinal reflex arc. What is that? Spinal reflex arc is, so there is an injury, somebody pricks it, immediately we move away from that injury side. So this comes, relays the information. From here there are two ways. It can relay the information to another one to take it to the brain, or immediately talk to another neuron that is there in the motor department adjacently. Got it? So immediately the arc is complete. Even though the information is relayed, it will talk to the motor neuron. So you are receiving information through posterior root and effecting an action through the anterior root. Most of the cell bodies and unmyelinated fibers are there in the gray area of the brain. It is the center, and the lateral part and anterior posterior parts, they are mainly made up of different kinds of tracts, which are nothing but again these nerve fibers. So three important takeaways. Gray matter is made up of neuron, white matter is made up of axons because of myelin sheath, and we call a nerve fiber nerve only in the peripheral nervous system, outside of central nervous system. If the same nerve is there inside central nervous system, we call it as a tract, not a nerve. So there are no nerves in central nervous system. And finally, collection of cells, collection of nuclei coming together and performing a function, a common function. It is called a nuclei inside the central nervous system. But if the group of cells lying outside of central nervous system, we call it as a ganglion. Okay, so this is the structure of our spinal cord, basic structure of spinal cord, and the white matter, as I said, is made up of axons, and these axons are called tracts. Okay, so now if we look at the nerves, how does our say this is the brain stem, and from here we have big spinal cord. Okay, this is big spinal cord, right? And we have different nerves going in from different parts of the body. These are called spinal nerves. They are called spinal nerves. These spinal nerves, we have different classification. What are they? We have cervical, thoracic, lumbar, sacral, and coccygeal. Okay, so you take a miniature of this. This spinal cord, brain stem. This has got nerves coming from all over the body. They are going up. We studied we studied here, right? The structure of a neuron and how it is is going into the spinal cord. From spinal cord, now we are going into the brainstem, right? So up to this we covered. So finally, the cervical nerves that are coming are 18 in number. Thoracic is 12, lumbar is 5, and sacral is 5, and coccygeal is 1. Total 31 spinal nerves from either side of the body are coming together, forming a spinal cord and going up and ending up in brainstem. So, what is the structure of brainstem? What are different parts in it? What is midbrain? What is pons? What is medulla oblongata? What kind of nuclei are there? What kind of functions they do? And from there, what is cerebellum? And finally, what is cerebrum? That's it. That completes the anatomy and physiology. So, simple logic. How a nerve functions is important. Once you understand that, it's all like coming together. How you are grouping different things. The same nerve is there in all the organs. Okay, group of nerve cell bodies. Group of nerve cell bodies is nothing but nuclei. There are different kinds of nuclei in our brain. Different kinds of nuclei are there in our brainstem. So everything is done. So in the first class itself, in the first half itself, we covered everything that is related to neurology. Okay. So what is the structure of neuron? What are the two important functions? Receiving information and affecting an action. So that forms the crux of neurology. If you go through it a bit more, time and again, a few times, the concept will be clear. Because now since we know all the cells, constituents of nervous system, we are just arranging them in a way that it makes a logical sense. So spinal cord is what? Spinal cord is nothing but the collection of mainly the tracts, the nerves, taking the information or relaying the information with a small grey matter which is nothing but unmyelinated nerves and some cell bodies. That's it. Okay. So we'll see and we'll discuss brainstem and cerebrum and cerebellum in our next class.